Hello everyone, this is second lecture on heteropolysaccharide. In previous lecture, we discussed about glycosaminoglycan, which are heteropolysaccharide present in vertebrates. In this lecture, we will talk about peptidoglycans, which is present in bacterial cell wall, and agar, which is present in some species of red algal cell wall. So let's first talk about peptidoglycans. It's made of two words, peptido and glycans. Peptido means peptides, which means it is made up of amino acid, and glycans means polysaccharide. Polysaccharide unit of peptidoglycans is composed of N-acetylglucosamine, in short we call it NAG, and N-acetylmuramic acid, in short we call it NAM. So, in peptidoglycan, polysaccharide unit is repeating disaccharide unit of NAG and NAM. So what is the difference between NAG and NAM? It's heteropolysaccharide, means it has two different types of sugar. So in NAG and NAM, both have at second position acetylated amine is present. Here at second position, this is acetylated amine. Here also acetylated amine is present. But major difference is at third position in NAM, we have lactic acid present here. So NAM is present in this box. We have this NAM structure. At third position, we have this lactic acid, which is absent in case of N-acetylglucosamine. With this lactic acid, tetrapeptide is present. So because of this peptide, this is called peptidoglycans. This is the peptide part of peptidoglycan. This is the structure of peptidoglycan. So this is the heteropolysaccharide repeat of NAM and NAG. NAM is attached with this tetrapeptide, which are present here, and two different heteropolysaccharide chain are linked through this peptide with this pentaglycine cross link. Pentaglycine means 5 glycine are present here. So how this glycine attach? So this third number of L lysine of this first chain attached with this fourth number amino acid, that is D-alanine of second chain. So they are forming the 3D cross link together, and they are forming the layer of peptidoglycans in bacterial cell wall. So this is the structure of bacterial cell wall. This gram positive bacteria and this is gram negative bacteria. They both have different peptidoglycan layer. In case of gram positive bacteria, this is cytoplasm, this is plasma membrane, yellow color, and this purple color is peptidoglycans. And peptidoglycan layer in gram positive bacteria is quite thick. It is of 20 to 80 nanometer thickness. In case of gram negative bacteria, here we have cytoplasm. This is in yellow color. We have plasma membrane. Then we have this periplasmic space, which is not very defined in gram positive bacteria, but periplasmic space is present, very well developed in case of gram negative bacteria. And peptidoglycan layer, if you look here, purple color, in case of this gram positive, this layer is very thick. In case of gram negative, there is very thin layer of peptidoglycan, which is only 2 to 8 nanometer thick. And apart from that, outer membrane is also present in gram negative bacteria, which is absent in gram positive bacteria. So this was all about the peptidoglycan. This is the peptidoglycan is made of peptides and glycans, and glycan layer means polysaccharide unit is repeating unit of NAG and NAM. Next one is agar. It is heteropolysaccharide present in some species of red alga. Examples are Gelidium and Gracilaria. Agar is made of two different types of heteropolysaccharide. One is agarose, which is unbranched and neutral polymer, which constitute about 70% of agar. Second one is branched and anionic component, that is agaropectin, which constitute about 30%. So this is the structure of agarose. Agarose has repeating disaccharide unit of D-galactose and 3,6-N-hydro L-galactose. This is here in case of 3,6-N-hydro L-galactose, this third number carbon and 6 number carbon are connected with ether linkage, and this D-galactose and 3,6-N-hydro L-galactose are connected with beta 1,4 linkage. First carbon of this sugar is connected with fourth carbon of this sugar. But in next sugar unit means this 3,6-N-hydro L-galactose connected with next D-galactose with third number carbon. So first number carbon of this sugar is attached with third number carbon of this sugar, means D-galactose. So they have two different types of linkage, beta 1,4 linkage here and beta 1,3 linkage here. And repeating this disaccharide unit of agarose is called agarobiose. So we can say that agarose is repeating disaccharide of agarobiose, repeating heteropolysaccharide of agarobiose. I have not shown here the structure of agaropectin. Agaropectin have same backbone structure of D-galactose and L-galactose, but major difference is it is branched and highly heterogeneous structure, and it is attached with many anionic groups like sulfates and pyruvate. So agar is used as surface for growth of bacterial and yeast colonies. So here this is the agar plate showing the growth of yeast, and bacteria are also grown in labs on the agar plates. Second one, if agar is polymer heteropolysaccharide which is made up of agarose and agaropectin, and agaropectin is highly negative, and when agaropectin is removed from this agar we obtain the agarose, and this agarose have gel forming property, as it is in labs agarose gel is used to separate the DNA and RNA through the electrophoresis. So this is the agarose gel, and it is the DNA present on separated on this agarose gel. So this was the use of agarose and agar in laboratory. So this was all about the peptidoglycans and agar. Thanks for watching. See you in the next video.